amino acid and protein part 2. In the last video, that is part 1, we have seen about what is the amino acids, what are the proteins, and how the amino acid get synthesized by different methods. Then we have seen deuterion, then isoelectric point, and how the peptide bond is formed. So, but now in this unit, in this video, we will see about the polypeptide bond formation and what are the principles which are using in the method of synthesis of polypeptide. So, we will see one by one. First point is synthesis of polypeptide, means we have to synthesize a protein molecule by linking two amino acids together, a liberation of a simple molecule and the formation of polypeptide bond. So, polypeptide bond is formed, and in this way the formation of big protein molecule. So, some important principles which are governed are very important for the all method which are applied for the synthesis of polypeptide chain. Number 1 is protection of amino group. So, the amino group means the amino acid contains amino group and carboxylic group. So, we have to protect the amino group, because if you react the carboxylic group, then we must protect the amino group, otherwise amino group and carboxylic group both are get reacted. So, for that we can use a benzyl chloroformate. So, the benzyl chloroformate molecule get reacted with the NH2 part of the amino acid, and a simple molecule HCl get liberated and formation of derivative of that amino acid. This is the first step, and the second step is activation of the carboxylic group. Now, we have to activate the carboxylic group by substituting the OH of the carboxylic group by the chloride atom, that perform when we can react with the SOCl2 molecule. So, that derivative of the amino acid which was formed in the first step, that react with the SOCl2, then you will get the chloro-substituted product. Then in the third step is coupling reaction. Now, we have to couple means together the two different amino acids to form a peptide bond. So, peptide bond formation takes place by condensing activated carboxylic group of the amino acid and with the amino group of the alanine. So, the previous formed chloro-substituted product is react with the alanine, then alanine molecule of the hydrogen from the NH2 and chloride atom of the first group chloro-substituted product get combined and formation of a polypeptide bond, CONH bond, new bond formation and formation of such type of polypeptide bond. Now, our aim is over. Now, we have to remove the protecting group. So, the first step is removal of the protecting group. Protecting group can be removed by reduction method, by applying the palladium as a reducing agent or catalyst. Then a reduction takes place and formation of glycyl alanine molecule. So, in this way, these are the 4 important principles on which any method can be based on these principles. Then we have seen the structural determination of polypeptide bond by N group analysis. What are the N group analysis? The peptide is the first hydrolyzed to its constituents amino acid, which are separated and identified. The amount of each amino acid is measured. Now, identify the sequence of the amino acid in peptide molecule. For that, the various chemical methods has been used to remove either of the 2 terminal amino acids residue, that is carbon terminal and nitrogen terminal, of the polypeptide in stepwise manner, so arrangement of amino acid in the polypeptide can be established. So in this way, by using this concept end group analysis, we can measure and we can determine the structure. So there are two methods, that is N terminal residue analysis and C terminal residue analysis. So first of all we see the N terminal residue analysis. So first method is Sanger's method. Sanger's method means, in this method we have to use the Sanger's reagent. So the reagent is 1-fluoro-2,4 dinitrobenzene. So FD, NB, see here, this is the reagent which reacted with polypeptide molecule. In presence of sodium bicarbonate, a simple molecule HF molecule, hydrogen fluoride molecule, get liberated and formation of derivative of 2,4 dinitrophenyl derivative of polypeptide. On hydrolysis, form DNP derivative of the N terminal amino acid. So dinitrophenyl derivative of the N terminal amino acid is formed and the rest of the amino acid residue. So in this way, different residue and the particular part which we have to determine the structure, that can also be removed. So in this way we can analyze by using N terminal residue analysis. First part is over. Second is advanced method or reagent. Advanced reagent is phenyl isothiocyanate molecule. So phenyl isothiocyanate molecule can be used for the reaction with the amino acid or polypeptide bond molecule. Hence the nucleophilic addition of NH2 group of the polypeptide to the carbon double bond nitrogen of that isothiocyanate molecule. In basic medium, in acidic medium, OH negative is there. The addition product undergoes ring closure, and after ring closing, the formation of phenyl thiohydantoin. This is the big molecule which is formed, and then that can be analyzed by the chromatography. So what are the process? The process is the peptide to be sequenced, means which the peptide we have to sequence, that we have to measure, we have to identify, we have to study that molecule to be sequenced is absorbed on a solid surface of the glass fiber. Adsorbed, not absorbed. Adsorbed on the solid surface of the glass fiber, which is coated with a polymer that is known as polybrine. Then advanced reagent can be added on that polypeptide molecule, and then the reaction takes place. That reacts with the N terminal amino acid to form cyclized product, that is phenyl dithiohydantoin. So in this way phenyl thiohydantoin molecule is formed, and that can be analyzed through the chromatography techniques. Now we will see the next point, that is the C terminal residue analysis. Up till now we have studied about N terminal residue analysis, and we have seen two methods, Sanger's method and Edmonds method. Now this is the C terminal residue analysis by using enzymes called carboxypeptidase. Here a special type of enzyme used for the analysis of C terminal residue analysis, that is known as carboxypeptidase. This enzyme hydrolyzes the peptide linkage at the C terminal, which holds an amino acids unit with a free carboxylic group. After hydrolysis, one product contains free amino acid unit of the carbon terminal of the polypeptide under analysis, and other is the rest of the polypeptide molecule. So in this way the rest of the polypeptide molecule is formed less than one amino acid molecule. So that's why in this way we can analyze, we can determine by using this method as C terminal residue analysis. So I hope this video you enjoy